Hello everyone, this is Dr. R. NITHYATHARANI, Assistant Professor from Department of Microbiology, Cauvery College for Women, Autonomous, Trichy, Tamil Nadu. At the onset, I welcome you all to the 3 days national workshop on CSIR net preparation in life sciences, organized by Botany for You. I am going to discuss about the microscopic techniques from chapter 13. The word microscope is derived from the ancient Greek, in which micros meaning small, scopian means to look at or to see. It is a laboratory instrument used to examine the objects that are too small to be seen by the naked eye. And microscopy is a science of investigating small objects and structures using a microscope. Microscopic, it means being invisible to the eye unless aided by a microscope. Anton van Leeuwenhoek, a Dutch businessman and scientist, is commonly known as the father of microscopy and microbiology, and he is considered to be the first microbiologist too. He discovered blood cells and microscopic nematodes and studied the structure of wood and crystals. He also made over 500 microscopes to view specific objects. He is best known for his work on the improvement of the microscope and for his contributions towards the establishment of microbiology. Let us now see as how a microscope works. A general biological microscope, it mainly consists of an objective lens, ocular lens, lens tube, stage, and reflector. An object placed on the stage is magnified through the objective lens. When the target is focused, a magnified image can be observed through the ocular lens. Thus, the magnifying power of a microscope, it can be defined as an expression of the number of times the object being examined appears to be enlarged and is a dimensionless ratio. It is usually expressed in the form 10x, which means for an image magnified tenfold. The resolution of a microscope is a measure of the smallest detail of the object that can be observed. It is expressed in linear units, usually micrometers. Numerical aperture is a dimensionless number that characterizes the range of angles over which the system can accept or emit light. Microscopes can be separated into several different classes. One grouping is based on what interacts with the sample to generate the image, that is, whether it interacts with light or photons, or with electrons or a probe, and is named accordingly as optical microscope, electron microscope, and scanning probe microscope. Let us now discuss about the conventional optical microscopy. This is an optical instrument containing one or more lenses that produce an enlarged image of an object placed in the focal plane of the lens. Resolution limit is submicron particles approaches the wavelength of visible light, that is 400 to 700 nanometer. Transmission is beam of light passes through the sample. Reflection, beam of light reflected off the sample surface. Microscope samples are usually fine powder or thin slices, that is, they are transparent. This picture shows the image of an optical microscope wherein you could see the ocular point, the objective lens, the stage, the base having the illumination control points, and the arm having the focus control. Next is the polarizing microscope. Here, only the light component whose vibration direction is parallel to the polarizer is permitted to pass through. The interaction of plain polarized light with a double refracting specimen produce two individual wave components. One is the ordinary ray and the other is the extraordinary ray. So these are polarized in mutually perpendicular planes. They are of different velocities and different propagation direction. Image showing the polarizing microscope. Let us now see about the dark field microscope versus the bright field microscope. The bright field microscope uses the normal wide field elimination method. The background here is bright and is low contrast. The characteristic feature of a dark field microscope is an opaque disc is placed underneath the condenser lens. It has scattered light, background is dark and is of high contrast, wherein we could visualize the structural details clearly. Next is phase contrast microscope. The field is bright and the special characteristic feature is that destructive interference patterns in the viewed image occurs. That is, the amplitude and phase difference occurs. Details in the image appear darker or brighter against the background. Colorless and transparent specimens such as living cells and microorganisms. So this is how the phase contrast microscope works, where you could see the phase ring. So next you have the objective, specimen, condenser, and it uses the annular ring. Next we have fluorescence microscope. Fluorescence is the property of some atoms and molecules to absorb light at a particular wavelength and to subsequently emit light of longer wavelength. So especially it is useful in the examination of biological samples. Fluorescence microscope has the following characteristic features. It identifies the particular molecules in complex structure, example cells. It locates the spatial distribution of particular molecules in the structure. Biochemical dynamics, high signal to noise ratio, both reflected and fluorescent light. The drawback is the use of chemical labeling. This image it shows the working of the fluorescence microscope, in which it uses the diachronic mirror and two types of filters are used. One is the emission filter and the excitation filter. The specimen is finally detected using a detector. Next is laser scanning confocal microscope. It scans a diffraction limited point of excitation light across the sample. The out-of-focus light rays are eliminated from the image by the use of a confocal pinhole. It is an important technique for live cell and tissue imaging. So there are a few advantages. First is the optical sectioning ability. Next, 3D reconstruction. It has an excellent resolution, specific wavelength of light used, and has very high sensitivity. So there are also certain drawbacks. It is expensive, it is complex to operate, chemical labeling, high intensity laser light. This picture shows the working of a confocal microscope. It uses the confocal pinholes where The out of focus light rays are eliminated using this. You could also see the high intensity laser beam. Having done about the optical microscopy, let us now see about the scanning probe microscopy. So here we'll be seeing about the scanning tunneling microscopy and the atomic force microscopy. Coming to the principle of scanning tunneling microscopy. So when a conducting tip is very close to a conducting or semiconducting surface and a bias voltage is applied, there will be a tunneling current flowing between the tip and the surface. The tunneling current is a strong function of the gap between the tip and the surface. If the tunneling current is monitored and maintained constant by adjusting the gap, the elevation of the surface can be traced. The surface morphology in atomic resolution can be obtained by scan. So using this scanning tunneling microscopy, we can determine the conductivity and the band gap. This picture, it shows the image of the scanning tunneling microscopy. The next we have the atomic force microscopy. The molecular force is a strong function of the separation between two objects. The force can be monitored by the deflection of a cantilever, which is in turn amplified by the deflection of a laser beam. Constant force is maintained by adjusting the position of the surface. This picture, it shows the working of atomic force microscopy, wherein you could see the use of a 100 to 200 mm long cantilever. So finally we have come to the electron microscopy. Yes, there are two major types of electron microscopes, that is SEM and TEM. So TEM is transmission electron microscope and SEM is scanning electron microscopes. They both have series of electromagnetic and electrostatic lenses to focus a high energy beam of electrons on a sample. In a TEM, the electrons pass through the sample analog to the basic optical microscopy. This requires careful sample preparation since electrons are scattered strongly by most materials. The samples must be also very thin, that is below 100 nm, in order for the electrons to pass through it. Cross sections of cells stained with osmium and heavy metals reveal clear organelle membranes and proteins such as ribosomes. With a 0.1 nm level of resolution, detailed views of viruses about 20 to 300 nanometer and a strand of DNA of about 2 nanometer width can be obtained. In contrast to TEM, SEM has coils to scan the surface of bulk objects with a fine electron beam. Therefore, the specimen do not necessarily need to be sectioned, but coating with a nanometric metal or carbon layer may be needed for non-conductive samples. SEM allows fast surface imaging of samples, possibly in thin water vapor, to prevent drying. This is a picture showing the working of transmission electron microscope. This slide, it shows the working of scanning electron microscope, where you could see the scanning coils are used to scan the surface. With this, we have come to the end of the session. Thank you.